All right. Hello, everyone, and welcome to our April Cell Modeling Seminar. Today, it's my pleasure to introduce Dr. Jacky Snoep, who is a professor at Stellenbosch University, University of Manchester, and the Vrije Universiteit Amsterdam. He's contributed great work to modeling diseases like malaria, HIV, and tuberculosis, and has also made considerable strides in standardizing the modeling process. He and his team have developed JWS Online and the FAIRDOMHub, which are both platforms that can keep our modeling processes open and transparent, which we're going to hear about today. So welcome, Dr. Snoep.
Thank you, Veronica. That is a very nice introduction. And, let me try and get the system going by sharing my desktop. And start the presentation. Yeah. So as Veronica said, I will try and talk a little bit about the integration of the FAIRDOMHub and JWS Online to show some workflows running from experimental data through model simulations to actual publication of models, and trying, which is always very hard as a scientist, not to focus on the science, but maybe a little bit more on the tools, how to make this possible. And I think specifically for the Center for Reproducibility in modeling, that would be an interesting topic, right? So I think it's key to make the modeling process transparent, so which I mean to make clear what you've done, and reproducible, because I think it's really a very bad sign that the vast majority of the models that have been published are not reproducible. You cannot reproduce the simulations that the authors have published, and that was one of the reasons we started JWS Online, to, if we managed to get these models to work, that we could bring them to the public in such a way that nobody else would have to go through the struggles of doing this again. So that was the simple start of JWS Online. We started this in 2000. And later on, we started linking with data and used model workflows. And then we linked to the FAIRDOMHub, which was then called the SEEK still, to really link the data and the models together to make reproducible simulations. So what I tried to do is walk you quickly through the two platforms, so JWS Online as a start, and then the FAIRDOMHub, and then really show some simulations and some of the problems that occur with reproducibility of models, and then some of the solutions that we've come up with. So JWS Online consists of two databases. One is a model database, so that's a curated set of models. So these models do reproduce what has been published in the literature, and we currently have some 600 models. And this is the best known part of JWS Online. And one of the key features that distinguishes it a bit from biomodels, which is of course a similar initiative at EBI in England, is that you can simulate these models quickly in a browser. So there is a direct link to simulations in JWS Online. And these simulations, that we also store these in the database. We currently have some 200 simulations in the database. So these are specific simulations to reproduce figures and papers. So I'll show you some of that, some of the aspects of these two databases during my talk. And the way to create these JWS Online simulations is setml scripts. So that's SED, it stands for Simulation Experiment Description, and it's a description in markup language. And these descriptions are stored in that database. So let me quickly show you a very quick introduction to JWS Online via a fairly simple model. Now I see all these. Let me see if I can move this a bit. Do you see this as well? Does this obstruct your view? Okay, so I put it a little bit on the side. So this is a schema of a simple pathway. And what we try to do is also annotate all these model files. So if you right click on any of these species. This is DHAP. You get an annotation what that species is with unique identifiers. And it also shows you the chemical structures, and it links to databases such as SABIO-RK. In Heidelberg, Germany, which is a curated database for model parameters. So if you, let's hope this works. Yeah. So if you click on that link, it will automatically fill in this search field, and it is a bit slow, I must say, and then give you a hit on all the enzymes that have that particular species. Similarly, we annotate the reactions. So this shows also what the reaction is, what the rate equation is that is used in the model, the chemical structures of substrates and products, and a similar database link. Now, the way to run simulations in the AWS is on the top. You see these different tabs that show model functionality. So you can do a time integration, and then you simply can select here, say time. Let's run this model from 0 to 100. Let's look at 2 species. And then you click the go, and then it will do the time integration of the model and show the species. So on the right hand, you have the control over the simulation. On the top, you have the choices for the different simulation functionalities. And on the left, you have the model descriptions. You can change model parameters, initial values, etc. So the functionality is fairly basic, because it's not the idea to put out a full-scale simulator, right? So there are very good standalone simulators. Herbert has developed some. We have Copasi, and we don't want to compete with a standalone server, with a standalone simulator on a server base, because clearly we will not be able to compete with very fast simulators. This gives you a basic tool to simulate published models. If you like the model, you download it in a number of different formats. I can simulate it on your platform itself. We have the ability to look in more detail in these models, and then what you will see here is the SBML structure of the model. The database we use is really, the schema of the database is really fitted to the SBML schema. And then you have the option in the top here to create a derivative. And what happens now is that whatever you do now will be stored in a session for your simulation. So you can now actually make changes. So you can look at the reactions, and you can say, I want to edit some of these kinetic rate equations. So now you can now code your heart out here and change these kinetic laws, save it. And these models will now be stored in your session model. So if you go to a model database and click on this session model, you see here now a Curial 3, which is now the model with the changes that you've made. We of course cannot allow it that you make changes to the model in the curated database, but as soon as you make a change in the model and you save it, it is stored as a session model. Now these sessions are browser sessions. So typically these models will be there for a few weeks, but then disappear. If you want the model to stay there, you have to ask us for you have to send us an email, and we will create for you a login, and then the models will be stored on the server for much longer. Right? So if you work with session models, you must take care that you afterwards download the model or store it in another shape, because they will not be stored. Okay, that was very quick, a little bit on GWS Online. I don't want to talk too long about this. I think this system is fairly well known, and you can look it up in the literature also how to use it. The idea is, of course, to make it so simple that you would not need any introduction, and there is a good help file too. Now, let me now introduce very quickly again the data and model management platform that we work with. This is the FAIRDOMHub, and this is a very big initiative. It's also been around for more than a decade, and it was initially created. Let's see if I can move this away. It's initially created for data management of very large programs. So these were mostly European systems biology projects where typically 10 to 20 groups work together in one program, and then there were about 20 programs in the whole network when we started with this. Now what we thought is to create something that gives access for everybody in the specific project to the data file, so that modelers and experimentalists can exchange data and keep track of this. But one of the biggest queries is how can we control access? So it was not so much that we could create a system that people have access to the data, but more how can the scientists control to prevent people from having access, because everybody was very worried to have all their data files shared. So the system has a very good access control. You can control who can see the data. And this of course makes sense that during the development of a project or as a project runs, you don't want to have intermittent files, make them publicly available. Now with a simple click, you can change access control to public as soon as you've published your stuff. The way to organize these files is a little improvement from just storing all Excel spreadsheets and model files on the hard disk somewhere. It's in a hierarchical ISA structure. And this sounds maybe terrible because ISA stands for Investigation, Study, Assay. It sounds as if it's a very locked down type of structure, but it's actually very flexible because you can decide for yourself what you call an investigation, a study, an assay. For me, for instance, the investigation is typically a publication. So every publication will be an investigation, and then a study is typically a figure in a publication, and the assays are the data files linked to that figure. I'll show you a little bit more later how these ISA structures work. But it's a very flexible and still hierarchical structure that allows good control even over larger projects. Of course, the storage should be safe, so there is a time guarantee. It's stored on a service in HITS, so that's the Heidelberg Institute for Theoretical Studies. And the time guarantee is that it's going to be stored there for at least another 10 years. And that's just the practical thing. You can't say to store it for forever, but it's going to be there for at least 10 years. That is guaranteed. And of course, it runs on backup servers there. It has versioning, so every time you upload a new version of say a model or a data file, it keeps track of all the older files. And the neat feature is that it has on-the-fly DOI generation. So you click a button and you have a DOI of your investigation and study at that moment. So the DOI is fixed then, right? If you upload a new version, the DOI will link to the version that was there when you created the DOI. But you can create more DOIs. It's basically a snapshot of the investigation at that time. But it's not necessarily an investigation. You can also create a DOI for every model or data files, and that makes it easy to cite a data file, because that is often difficult. How can you how can you give a reward for people creating these data files and making them available in your publication? It's very hard to refer or to cite a data file now, but with the DOIs, this goes fairly simple. I will also talk on the setml later, because that is for us a very practical tool to store simulation experiments. Let me show you an example of such an investigation, and I show here an example of what Veronica referred to as our work on malaria. Let me collapse this a little bit to show the structure. So this, at this level here is the, can you see my mouse on the screen? Yeah, okay. So this is the investigation, and then these are 3 studies: model construction, model validation, and model analysis. And there are some publications linked to this investigation. If you open up the model construction, you will see all of the different enzymes. In this case, it's a glycolytic pathway, so I think about 12 enzymes. And for each of these enzymes, there is a study. So the hexokinase is the first enzyme. And then you can click on this, on the, say, the hexokinase kinetic assay. If you double click on that, it should go to that kinetic data. And this is typically an Excel spreadsheet. That's what we saw is the vast majority of experimentalists want to work with Excel spreadsheets. And the problem with spreadsheets is that it's not so easy to annotate them. So we created a tool. It was created at Manchester, a right field and tool that makes it easy to create templates so that you can annotate Excel spreadsheets. So here is an example of the metadata of one spreadsheet where you typically, in this case, it's a template for kinetic data. And you just give some specifics of how the experiment was done, and you also can define the unique identifiers for the reactions and the metabolites. And the data is a typical Excel spreadsheet data file. Now you can, of course, download that file. You just click on this button and it downloads the file. Then I create here a hexokinase model. In this case, I do most of my simulations in Mathematica, so I create a Mathematica notebook, but you can upload whatever model file that you've used. This can be a Copasi file or a MATLAB file, anything. And I'll quickly download this to show how I set up these files such is that if you open up that notebook in Mathematica, and you evaluate that notebook, it reads in the data file if it lives in the same directory of your notebook. You see here a rate equation, it parameterizes the rate equation, and it, in the end, reproduces the figures in the paper. So, let's see. Okay, well, you believe that, right? So the script is such that it reads in the data, parameterizes the rate equations. You find the same parameter values as published in the tables in the paper, and also the figures. So that is a bit primitive maybe, but it at least shows you how the figures in the paper were made, and you can redo the parameterization. You can, of course, decide, I don't like that rate equation. I'm going to use another rate equation and then fit that on the same data set, and maybe you get a better fit if you have a better idea on the mechanism of that enzyme. So that's done for all the enzymes as shown here. Then we have a model validation. And whereas the model construction was done for the isolated reactions in the system, the model validation is done on the intact parasites. So the malaria parasites were lysed to characterize the enzymes, and in the validation we incubate an intact parasite with glucose, and we look at the in this case, a glucose incubation. We look at a glucose run-out experiment where we incubate the trophozoites with glucose, and we take time samples. We, of course, have a kinetic model. And then let me show you here the first example of such a setml simulation. I've just copied that here in the description. If you click on that simulation, it's going to now load the model from JWS Online, take the data from the FAIRDOM, and publishes them together. So it has an instruction on how to simulate the model and how to plot that together, as shown here. That was quickly on some information on the FAIRDOMHub. So it's a very good tool to structure the data, and you can. And I focused a little bit on the workflow that we use, right? So it's model construction, model validation. Make the data that you've used for each of these different components of the modeling workflow, make explicit what data you've used, how for the model construction and the model validation. Okay, now let's talk a little bit about reproducibility and how we can now link the FAIRDOMHub and JWS Online. And for me, the key for this reproducibility is like say, I read a paper, and say I found a link here to this microbiology paper. Let me download the PDF. And I start reading this paper, and I find, okay, this looks all interesting, blah, 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 some NMR spectra, and then I come to this figure, and I say, oh, this looks fairly simple. I have a VPG, that's 1,3-bisphosphoglycerate. and it's dephosphorylated to 3PG. So it's a very simple reaction. I'd like to just show a simple example to show the strength is shown a little bit easier with a simple example, that you don't get confused with too much detail. What I would like to have is that for each figure you have this link. So you click on the link, and what it'll do is it reproduces the figure in the paper. That is for me reproducibility. It shouldn't be a big effort to load a model somewhere, figure out how to simulate the model, then integrate data with it. It should all run with a simple click. Ideally, I'd like to just click on the figure and then reproduce this. And don't get confused. This is not a figure that is downloaded, right? This is a live simulation. So if you click again on that create derivative, it tells you this script, it simulates a Coral 5 model from JWS Online. It takes the data from the FAIRDOMHub. It runs a simulation from 0 to 5.1, and it has some more information on what integrator to use and all these standards. And then it creates outputs. And those outputs, they have like data and model simulations. And we will take a closer look at how to generate all these scripts just now. I think it's nice maybe to just show that this is really live by just editing one of the kinetic parameters. You can say this is the degradation constant was 0.7. Let's say we make it 0.2. You save this, so this is stored again as a session model, and then you simulate it. What you see, it's still going to draw in the data from the FAIRDOMHub, but now with the lower degradation constant, the fit is of course not so good anymore, right? So it's really, this is live, a live simulation, and you can play with this. If you would now go to the simulation database, session simulations, you see there are now, I've created some variants of the original file in my own session simulation. So that's I think a key, right? If we can publish all our work with model simulations and data in such a way that we have such a link, there would not be an issue of reproducibility anymore. You just click on the figure and it should reproduce the figure. And you can still see how sensitive that model simulation is for certain parameter values that you want to test, maybe. For reviewers, this is of course also perfect to just play a bit with the model and see how does it behave. Now, next question is of course, how difficult is it to make such a thing? So let's look at that. So again, we go to the FAIRDOMHub because we will need some data in the FAIRDOMHub to download. So I've looked now at the investigation that I've downloaded the data from for the setml script. So we look at BPG stability. So you can look at this degradation, and again there is here so a BPG degradation Excel spreadsheet. So you can click on this. And well, it's much the same as we saw before. It's just a right field annotated Excel spreadsheet. But what's now nice is you can download it, of course. But now the key is now to just click on the down and copy the link. So that's the link to that data file, right? So in what we're going to do in the setml, we're going to copy this link so that we immediately refer to that data file in the FAIRDOMHub. So that's a nice to have link. The next step is simply to run this model. So for that we go to JWS Online, and this model was it was a model created by Coral. Sorry for moving this stuff around, but that's always in there. So if you can also search for organisms or processes or type of models, but if you know the name of the last name of the first author of a paper, you can immediately look for this here, and it was Coral 5, it was listed in the paper. So what you can do is just click on this green button and it will simulate this model. And you see it's a very simple model. It's just basically one reaction. Now what key now is to reproduce that figure, and that's a very simple thing, because the only thing you had to do for the model simulation is change the end time of the simulation and go, and this is basically the model simulation for that figure. Now we need to get the setml script for that. And that is now one of the most difficult tasks here, because you have to press this button, add to simulation, add to simulation. And now you will find in your simulation database a CUDL 5 experiment. So you could now simulate that, and you're going to reproduce that simulation, but it's now more fun to look at the setml script, of course. So the models, of course, refers to CUDL 5, data description. Hey, it's not there, because we just ran a model simulation. So what we do is we add a data description. And here we're going to give this an ID. So let's call this BPG degradation. No, that should go there. Sorry. Give this an ID. I'm not interested in human-friendly names. The source is going to be that. No, I know the stupid. I copied. Right. Sorry. I have to go back here. Copy that link back. So this is the link from the FAIRDOMHub, right? So because you, of course, not going to remember this. So if you copy the link from the download of your data file, and you copy it here in the source, that's a direct link to that data file in the FAIRDOMHub. Now we have to create some links to the data file itself. So we need an ID, so we want to have time. And the index set now, and this is a bit primitive, so I'd like to hear responses. We do this now, it's a bit of a hack. You refer to the first row of the column in the Excel spreadsheet. And it works. So even if someone inserts columns, it's going to still refer to the correct column, but it's primitive. So we'd rather use some sort of unique identifiers and then spacing in the Excel spreadsheet. So I'm open for suggestions how to do this. I know these headers now. So the time header was time. And I'm going to create another ID BPG. And I now happen to know that the first row of that column was BPG. I'm going to create one more entry, and that's the other metabolite, P3G. And that was called 3G. And that's it. So now this is a bit painful, because you need to know what is the first row in my Excel spreadsheet, but also not such a big issue. You save this. And now we have our data description. Simulation task is already stored because we did the model simulation, right? We did that in JWS Online. So the model part is covered. And now we have to do the outputs. Now we have to say what we are going to create, and what you see is what is our simulation output. The simulation output is already there for the model, but we need to create now the data files. So here we now need to give this an ID. So this can be anything. Plot data PPG. And on the x-axis now we have to say, okay, this is not FATA. This is from the data. And I'm gonna have now the ID for the data was PPG degradation time. That's what I want on the x-axis. And on the y-axis, I want the same data. PPG degradation. And I want to see PPG. So the data construct here refers, because it's experimental data that you plot. You see for the model, you have symbol for the x-axis and the species for the y-axis. Those are given. And this is the identifier in your data set, and this is the name. Okay, the next one is gonna be plot data 3PG, and we do data BPG degradation time, so it has the same x-axis, of course, and the y-axis is data BPG degradation B3G. So this was the identifier for the data set. This is the identifier for water on the plot. We save this, and let's hope I didn't make any typos. It looks okay. And then you can simulate this. And now it's going to redo the model simulation, but it pulls in now the data from the FAIRDOMHub. So fairly easy, but a bit, you have to hack a little bit how to get the data in there. What is nice is that if you have that simulation database, it has 200 examples of how to do this. So you should be able to figure it out by just looking at the examples, and looking at the details of these setml scripts will give you the information how to link data and model simulations. Okay, that was a little bit on model workflows and JWS, how to integrate setml.
Sorry, can I ask a question?
Sorry, yeah.
How does JWS Online know to use the right colors that match the experiment to the simulation?
Yeah. So that's when we started with this, there was no way that we could describe this in setml. So we of course we have complete control of the background, the mathematical kernel that does all the simulations. So we coded this such that it works.
So I can add something to that. So just last week, we approved the next revision of setml, which includes as many colors as you want. So in theory, you could do the color specification and a bunch of other things as well, line styles, markers, etc., etc.
But so what I meant was that the data set that you added to the simulation was like the species had the same color than the simulation, the simulated species. Was that? How does this magic happen?
It's not a trivial thing. I don't know if Davi is still listening. Davi, are you listening? Because I think you organized this. No, I think Davi.
Yes.
Okay.
I am. So at the moment, the coloring is just done by picking from the same color palette in the same order. So in this case, because you've got two simulations and two data sets, they happen to have the same colors. But yeah, at the time that we implemented this, there wasn't a specification, as far as I know, that you could use to uniquely label things with colors so that they would agree.
Mm-hmm.
So the other way to do is to match basically this entry in the data set corresponds to this simulated species. So at the moment there's nothing that says that the whatever it is BPG in a data set is the same as the simulated BPG, right?
Yes.
Correct.
Yeah.
Correct.
Okay.
But
so at the moment that interpretation, the correspondence between the data and the simulation would only be evident if you labeled your plot with a figure label on the side. So the names that you put into the output description, the names would appear in the figure legend.
Yeah, so that is, I was a bit quick, so I didn't type in the human readable thing, but if you type those names in, they are listed on the side, and then you see what the experimental dots are and what the lines are in the simulation. But of course, ideally what we want to do is what we started is to list the species with the unique identifier in the Excel spreadsheet, and then you want to use the same unique identifier in the model, and then you know what belongs to what. Yeah, but that's in the making. We don't really know how to. We now list those identifiers in the metadata sheet, but that's not ideal. You want to really link it to the data in the data file itself. But it's good to hear from Herbert that there is now actually also a way to define the colors, because what you saw in the first setml scripts that I showed, there was not a good link, right? You saw that the data and the simulations did not have the exact same color.
So one question, Jacky, I didn't catch. How did you go from the PDF file to the to the simulated graph. How did that happen? That was a bit of magic there you did. How did that happen?
Yeah, no, because there is a link in the. Let me see. How can I now go back?
I mean, you had to do something to the PDF file? Did you? I mean, are you storing the PDF file on JWS Online?
No, 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 no.
Oh.
So this is a link to the. Why is this not working as a link anymore? Oh yeah, right. Here. So here is. So I downloaded the PDF here on the. This is microbiology, right? The
right,
journal.
Yeah. Okay.
Here's the PDF.
Yeah.
And then you go to the figure, and what we did is we just in the figure legend. This is the link to the.
Oh, sorry, I missed that. Got it.
Yeah.
Got it. Okay, good.
It's a bit clunky. Of course, you would just like to click on the figure, right?
No, that works.
But the editors don't want to do anything to the PDFs because that's just too much work.
So somebody has to curate this then. Do you curate this then?
Yes,
okay.
Yes, and some students in my group.
Got it. Okay.
But I made these myself, so these were easy to curate.
Yeah, this is workable. Yeah. Okay, nice.
But it's quite a bit of work, of course, to do this, but it is really worth the effort because you immediately know whether or not it's reproducible.
Yeah, that's nice.
Here, right? Okay, so this is, of course, one of the keys, of course, is that we do a lot of this work together with journals. So one of the top journals that we work with now is molecular systems biology. And I just created so there's a journal program on the FAIRDOMHub, and then for every paper that I curate the model for is we just make an investigation and link the data files there. And this is then, and here the curation that we do is technical curation, right? We don't look at the science, we are not the reviewers. We just make sure that the model simulation runs and reproduces the simulations in the paper. Okay, so these are some, I think, key aspects of making models and model simulations reproducible is to make the workflow for construction and validation transparent. So most of the time people say we constructed the model and we validated the model, but they don't make explicit what data were used where, how did they parameterize how did they get parameter values, what scripts did they use for identifiability of the parameters and stuff like that. Yeah. We think it's key that validation and construction should be separate, and you should make explicit what you did where.
Yeah, great.
And then make the data model files that you've used available. And there is no excuse not to, right? There are very good tools to do this. And I think standards are key. So for the models we used SBML, but there are, of course, other model standards, like setml, for instance, is also a good description if you work at the higher hierarchical level. And setml for the description. So there are just rules how to write these files, and because there are these rules, many tools can use SBML. So for instance, Herbert's Roadrunner can work with SBML, but Copasi works with SBML, we work with SBML, and that's nice to compare model results with different tools.
So Jacky, so a question for you. I mean, I see so many models coming out in MATLAB. I mean, how can we move people over from MATLAB to use standards?
Yeah, so there used to be a package that you could, of course you had to code your MATLAB file in a certain structure, and then you could save an SBML file.
Oh yeah, I know the one you mean.
That worked on that is not updating it anymore, but that was helpful.
Yeah.
But now, so what we often do if we work with these models, we get the MATLAB files from the authors and we just code it in them. And so we use our JWS description and create the SBML files for it.
Oh, you reverse engineer the model?
No, we get the MATLAB file, so we have it in electronic form.
Yeah.
And then we extract the ODEs from it.
Oh, just the ODEs. You don't get the network.
No, we don't.
Okay.
But it is it's not often not trivial because these MATLAB files are often very, you get 20 files and then it's like sort out what is what. Yeah. So it's not trivial. It is a lot easier if people work in SVML.
Yeah.
But coming back to Herbert's question, how just getting the ODs out of MATLAB is really not very useful if you want to move forward and we use their model and extend it and change it. How do we get people to stop doing that and actually describe the semantics behind their models?
Yeah.
Yeah, so I think a key would be, of course, to get the journals to adhere to these modeling standards, right? So every researcher wants to publish their work if they find out, oh, we can't just dump our MATLAB files anymore. We have to actually work a bit to get a proper model description.
Yeah.
That would help, but it's hard because the journals don't want to make it harder for themselves that they actually have to check these models, and they don't want to make it harder for the authors to submit papers. Yeah. But this is a difficult one. But it's much better for the model descriptions than for the data files, because it's very rare that you have actually access to the data files. This is just supplementary material and you get the PDF, right?
Yeah.
And so you don't get the data in electronic form at all.
Yeah.
And that's much harder. And also there are no clearly defined formats. It's just Excel spreadsheets with data everywhere. Right? I see much more problems with the data than with the models. With models, if you look how much progress we've made in the last 10 years, I think that will sort itself out. The data files is much harder. Now, before I completely lose my voice, I'd like to finish, and I'd like to show one last thing, because, well, of course, I'm as bad as these MATLAB files, because I code in Mathematica, but at least I make my files available. And I make SVML files, but now I just don't seem to be able to get out of this screen. How did I do that just now? Sorry, I'm struggling a little bit to get out of the Zoom. Anyone has an idea how I switch off?
Are you trying to get out of the screen share?
Yeah.
Okay, I can manually stop that, I think.
You can do that.
Maybe.
Interesting.
Okay. You should be out now.
Yeah. Now I can actually go to another program. And now I should be able to switch it on again, right? Let's share. Oh, the host has disabled. Can you enable me again?
Okay, you should be able to now.
Okay. Yeah, there it goes. So that, share. What I'd just like to show is just a few examples of how we work with, say, the FAIRDOMHub and our own models from a more technical point of view, that we don't want to click too much in browsers, and we rather work in coding environments, right? So I show one example of how we do that in Mathematica. So we've written some packages, that's the simulation packages that we can easily run simulations. And then to access the API from the FAIRDOMHub, you basically, in Mathematica, you just define the URL, your username, your password, and then I run a URL execute. Now, if I run this, I'm now logged in the FAIRDOMHub, and I can just do a simple query. So this is a simple query. How many papers do I have in the FAIRDOMHub So and it's very easy to define these things. It's a search, you can define your query, I look in publications, and that's it. And it's all JSON files that you get back, so you can immediately use, in this case, a data set structure that shows nicely how how you can work with those publications. Now, more useful maybe is that I can also say, okay, get me that same data file that I've worked with. So now I just say, give me some data. This is the link for the download, and it's an Excel spreadsheet, an XLS format, and here I define which of the sheets I want to see, and I want to see the last one, so that's here. And then I can, of course, make a plot of the data. Now here, I use our REST API for JWS to get a model from JWS. So with this get JWS model, that's the function defined in the packages. I load the Curial5 model, I do a time simulation, and I plot this together. So what you see in this coding environment, I have direct access to all the models in JWS and to all the data files in the FAIRDOMHub, and I can really make workflows, right? So if I just change the model name, of course, I can run the same script for another model. I think this is, the web browsers are nice to look at and to show how the data is structured, but if you want to do some programming, you want to do it in a workflow. And I think the API that we have now for the FAIRDOMHub and the REST interface for JWS Online make that possible. Good, and I think that brings us to the end. Let me thank some people, because of course we did this with many people together. So the setml script was done together with Dagmar Waltemath in Rostock, and she had a student, Martin Peters, come over here to work on the simulation database. We have a fairly big team in Stellenbosch and also some people in Amsterdam to work on the JWS Online database, and this is just the current setup, right? I can't list everyone that worked on that for the last 20 years, but students helping in the curation is a big deal. Curation is still a manual job, and that doesn't make it easy to scale. The FAIRDOM team consists of two parts. That's the Manchester team under Carol Goebel at Computer Science, and the people at HITS with PI Wolfgang Müller. That have also a big team, and also some of these people work on the Sabio RK database that I've shown. And some of the experimental data came from the group of Tina Siebers in Essen. And David, I'd like to thank you for your attention. I hope I have left some time over for discussion, but it should be good. Thank you.
Thanks a lot.
Okay, we have time for some questions. Would anyone like to begin? Arthur.
So I'd like to ask you a very nice talk and very nice work. And I'd like to ask you a question about problems of scale. You know, some models, of course, are small, like the ones you showed, but some are quite large, and they involve a lot of data and multiple simulation runs, which may involve a lot of computing. And how would you suggest that reproducibility be scaled to those larger, more complex models and simulations?
Yep, very good question. And indeed, I showed basically some simple models to make it more illustrative. But the problem becomes, of course, much bigger if you have really, say, thousands of metabolites. And just figuring out what metabolite you're looking at is then already a big issue. For instance, the schema that I showed, the rendering becomes a big issue if you have genome scale models. I have no easy answer for it. So running the models is not such a big issue. I think we can run ODEs, not necessarily in your browser, right? So JWS Online is not very well suited for kinetic models with more than, I would say, 100 reactions, then it just takes too long to run it in your browser. You're not going to wait for more than 2 minutes. But I think there are good tools to simulate big ODE models. But how to test for reproducibility with thousands of metabolites? I have no answer for that. That's a big issue.
I think certainly one direction we need to move is to make it more offline rather than online. I mean, I think we still want to be able to achieve reproducibility, but it's not going to be as instantaneous as the examples that you showed. And so it's going to need to involve, you know, just adequate access to data and compute resources in order to be able to get the results, and it will take longer, but that doesn't mean it isn't reproducible.
No, so I definitely agree. We have to go offline if the models get really big, because it doesn't make sense to do that online in the browser or on the server. But I see the problem more in developing good software to check, right? How you going to keep track of 1,000 metabolites in the simulation? How do you link it, the model simulation, to the data, and how it was published, and how is it reproduced? Yeah. I think that's probably more key than to simulate a couple of thousands ODEs.
And in a sense, there are two problems here. One is for the more common model, which tends to be small, and this kind of environment works quite well. And then there's a growing number of very large models where perhaps we have to think of a different approach and an offline approach. So there's probably a case for approaching two separate problems in different ways, because we still we'd still like small models to be immediately available to people and not batch it. But for large models, yeah, you'd have to start batching it.
So an approach that we've taken for if we go to larger scale systems is we use a modular approach. So we make multiple modules for smaller system and then link them after they've been validated. We don't make parameter changes on those modules anymore, and link them then afterwards to get to bigger systems. But those are still not thousands of reactions. It's maybe 100 reactions.
But this problem even arises for smaller models, actually. If you do a parameter estimate, parameter fitting, on 30 species, right, with a large amount of data, you could be running it for days.
So what we try to do the parameter estimations, say, on the isolated components, right? So what I tried to show with the malaria model is that you fit it on the isolated reaction, and then you don't change the parameters anymore.
Oh, I understand. Yeah, of course. But a lot of people don't do that still. You're one of the few groups who does that.
Yeah, but maybe it's also a bit dependent on what system you work. Metabolism, okay, you can measure good kinetics, you get good rate equations, and
I think it's the right. No, I think it's the right approach you're taking. Yeah.
I'm not sure. I think the other approach is you can't even identify the parameters, right? You must have such a detailed data set to have 30 parameters identifiable. That's very hard.
Right. Yeah, right.
I have one question. I'm wondering about how, I guess, documentation works from the actual experimental data into numbers that are used in the model. How is it documented, I guess, what particular number was used, if maybe there's more than one data point, or how it may have changed during the parameter estimation? Is there any documentation on that? I guess if there are any efforts going forward.
You broke up a little bit. What I heard is that how to treat multiple data sets, or
how I said, can you hear me now?
Yeah.
Oh, how the documentation works for how maybe a modeler selects a single data point if there are many experimental values, like how it gets estimated, or maybe how the numbers change after parameter estimation in model construction.
So what I tried to show in that in the first model I showed with the malaria model, right? So you see the complete data set that was used for the parameterization of the model. So all the data files are there, and there are multiple. So these are big spreadsheets, right? So typical, if it's a two substrate, two product reactions, you have at least 4 variables in the spreadsheet. These are varied independently, and then for each combination, you get an activity. But that data is gathered over a 3-year period. All right, it's a lot of data points in there. Also, so the variability there is mostly what we see is that if you do an experiment a half year later, these parasites, they might have been harvested a little bit later. You get differences in expression levels. So what you will see is in the paper there's a whole distribution of Vmax, so expression levels of the enzymes with standard deviation, because the Vmax has the biggest standard deviation in the different experiments. The Km values don't change so much, but the Vmax values do. But we published that all together in the paper.
I guess what I mean to ask is, oftentimes there'll be a difference between the experimentally measured value and the actual number that gets used in the model, right?
No. So we use experimental data to parameterize the model, right? So a model parameter value is fitted on the experimental data. I seem not to catch your.
So I guess maybe I don't know, Yosef, if this is exactly what you're trying to say, but like one example might be if you had experimental data and you performed like a global fit, so it didn't necessarily directly integrate the experimental data, but some fit to like multiple data points, and which could possibly vary based on how you do the fitting.
Yeah, that would be one example. Or another one, just maybe if you're trying to fit the parameters of your model, you need to vary the experimental parameters slightly.
Yeah, so what you can have is, so say you create, so what we try and do is we parameterize the model on, say, initial rate kinetics of isolated enzymes. Then what you sometimes find, if you add these enzymes together, then you don't get the perfect description of the complete pathway on the basis of an individual kinetics. So what we typically do then is we check, can it be, so of course the initial rate kinetics, you might not have checked each of the intermediates of the pathways, right? So there can be an allosteric regulation of pathway intermediates. But sometimes it's also that you find that an enzyme doesn't seem to behave exactly the same in a couple of hours incubation of a conversion of a substrate to product as it did in an initial rate kinetic experiment, which typically takes a few minutes. And then you have to figure out what parameters. So if you have a bad prediction of such a pathway experiment, we typically look at how sensitive, what's the sensitivity for the different parameters to get a better description, and can we pinpoint at a number of parameters that are off for some reason. But we try to go back then to the initial rate kinetics. Can I figure out what is going on? But sometimes you just don't find out why that one parameter behaves differently. I think the key there is to document. So we document this stuff in the paper. We say, okay, this is the value we used that we got from the initial rate kinetics, but the model does not predict the data set correctly. But if we change this one parameter with 20%, it does. Then if we can't figure out why that is, then we just write it down. Sorry, we don't know why that enzyme behaves different in the pathway. Is that more what you? Yeah, okay. It doesn't always work right now.
Yeah. Okay, well, I think it's time to end the seminar for today. Thank you so much for this talk. This really was incredible. It's like such an intuitive way of linking, data from the experiments to actually simulating the model. So thanks. This was really interesting for me. And everyone, thank you for attending the seminar. This is the last one of our first series of seminars, and I'd like to just quickly introduce what will be our next series. The overall motivation for these seminars is to, sort of create a virtual community for everyone working on wholesale modeling, because we're, so physically distributed. This initial series, the main goal was to sort of establish the abstract methodology and goals of wholesale modeling, particularly what challenges we need to face. The upcoming series, which is going to take place over the summer, is going to be focused on, actually implementing new solutions. It's going to be mostly, showcasing new software that was made and tutorials for how to actually use it. We think this will be, a crucial next step for forming a community, and that we'll actually discuss new technical solutions that have been proposed. So I'll send that email about that shortly. If anybody, would like to volunteer to give a tutorial on any new software, I'm going to add in my email in the chat. The seminar email. But yeah, please reach out for any suggestions. Otherwise, look out for an email over the coming month from you guys at the new seminar. Thank you everyone for attending.
Okay.
Thanks a lot.
Thank you.
Bye.
Thanks, Jacky.